Hello, SEA-PHAGES. Welcome to another week of pre-open lab videos. I'm going to be going over three-phase streak today. We're going to be doing this protocol for 3 weeks, but it's important that every one of those weeks you watch the videos, okay? Because some things might be a little bit different. If you want to follow along, pages 33 to 34 in your lab manual, as well as 105 through 109. The 105 through 109 is where the specific streak protocol is going to be. So if you're wondering where it is, it's all the way in the back of the manual, okay? So just thinking holistically, where are we in the process in our phage discovery journey? So we last week isolated a sample via direct isolation, and then we're going to spend the next couple of weeks purifying our sample, really to make sure that we only have one species of phage, okay? So let's talk about finishing up direct isolation protocol. So first thing you should be doing when you're coming into open lab this week, outside of, wiping down your lab bench and putting on your lab coat, all that good stuff, is getting your plaque assay, direct isolation plaque assays from the incubator, and then looking for results. You should be taking photos from last week and putting them into your lab notebook, uploading them into your lab notebook that day. So these pictures for your plates are going to serve as your results. Okay, and these are going to be the results that you interpret in your analysis and your discussion and your next steps for your lab notebook. If you got negative results, and what I mean by negative results is you got absolutely nothing on your plate, these still count as results, right? There are a lot of things that could have given you those results. It could have been that there are just no phages within your sample that could infect Gordonia terra. Could be something contamination-wise. It could be other things going around in your plate, like some fuzzies growing. Right, all important things to note. So consider all those factors together when you're thinking about your analysis, right? Your results, it's just the photo, what it is, brief description, measure the plaque sizes, describe them. Are they cloudy? Are they clear? Things like that. But then analyze that. So what's going on your plate? The size, are they consistent sizes? Are they all the same? Are they all different? What do they look like? What's the morphology? What do you think the morphology means, right? So if they're clear plaques, most likely they're lytic. If they're cloudy, most likely they're lysogenic. So what's going on, what's going on with that? Describe what you see and what that means. And then for our next steps, we're gonna be doing our purification steps, okay? That's our next steps. So explain what you're doing and why you're doing it. What are you trying to purify? So just a recap, right? What was direct isolation? What did we do there? So it was a quick method to detect the presence of phage from an environmental sample, from our soil sample, but it gave us a snapshot of all the phages that could possibly infect your host, right? You could, hypothetically speaking, have a bunch of different phages that could have infected Gordonia in your sample, right? So if you're just looking at your plaques on your plate, how do you know if that's a single species or if that's multiple different species, right? You have no way of knowing. So how do you ensure that there's only one species present? That we only want to isolate one and characterize one? What are we doing to do that? Okay. This is where purification comes in. So let me give you an example with why it's important. So here on the left, I have what I would call an impure sample with two very distinct plaque morphologies. So what I mean by that is you have these little tiny dotted ones, they're really small. You have a bunch of them, a lot of them are consistent in the smaller size. Then you have others that are bigger plaques, right? Larger, and they're also, you see a bunch of them very consistent that they're all around this same size. So if I'm looking at that, I can't tell if that's one species, that is just some of those plaques are growing a little bit slower, or if that's two different species of phage that have different burst sizes that are maybe, quicker in infecting, things like that. I'm not really sure. So how do I know? What do I do? So what I could do is I could streak each one of those plaques onto a separate petri dish with, agar bacteria, streak them separately, plate them separately, and see what kind of results I get. So if I isolate this plaque here on the bottom, right, I purify it, I plate it again onto a brand new plate, and I only get this type of morphology, what that's telling me is this is its own unique species. This plaque here that I picked from is its own species. Again, if I took this top plaque here and I cultured it, purified it, plated it on a new plate, and I got plaques that were all, again, this consistent size, they all look the same. What those results are telling me is that my plate here on the left originally had two different species of phage that could infect my bacteria. But now that I've purified them onto their own individual plates, what I'm learning from that is that these were two different species. Okay, you're not seeing a mix of morphologies on both of these plates, right? That's where that mentality comes in. So we're going to purify a single plaque with the intention of by the time we're done with our purification, which is going to be 3 steps, 3 weeks. We'll ideally only have a single species of phage, which we are going to then further characterize to add to that SEA-PHAGES database. So this is the 3-phage streak. This is the protocol. It is a technique that is used to isolate a pure phage population for the purpose of characterization. You can think of it as a way of just repeatedly diluting your phage sample until you obtain a clonal phage population. What I mean by that is when we say clonal, like clones, they're all the same. So all the plaques and all the phages in your sample are from one species. Okay. And again, we do this over a series of 3 weeks. So we take this on the left, which you could call a mixed morphology plate, and we do this streaking process where we streak it across 3 different sections. That's why it's called a 3-phase streak. And we're essentially diluting it as we move down into those from section 1 to section 3. And I'll show you how all this plays out. So this is also another way of thinking of dilution. So on the top, you have the traditional 3-phase streak method that we will be essentially doing, right? But if you look on the right here, it's also very like, mostly think of it as for visualization purposes when I say dilute. So you can see on the bottom, this is a representation of plaques that would grow on this plate based on this dilution step. So as we move from section 1, this is going to be your most dilute area with the most plaques that will form. Section 2, a little bit less, but still highly concentrated in plaques. But as you move into section 3, you can see they're isolated. They're farther apart from one another. You can actually count the number of plaques in this section versus number 1. You can't really count it. So you're diluting the number of plaques as you move from section 1 to 3. And then on the right, again, this is just a visualization to understand the dilution process. Up here on top, where the streaks are really close together, really clustered, there's a lot more plaques. And as you widen those streaks out, you see more countable, more isolated plaques. So that's what I mean when I say dilute. So this is now same thing as the previous slide, but this is what they would actually look like. Okay, so your first streak area is going to be mostly lysed, right? This big clearing, it's not a mistake with the auger, nothing you did wrong here. This is just all the bacteria died, okay? And as you move into the second section, you can see, again, big areas that are just cleared off, completely lysed, but you can see the remnants of little round areas that tell you this is from plaques, right? You can see a couple more distinct ones. And as you move into this third area, very dilute. You have 1, 2, maybe I wouldn't even count this in the third section. You have one single plaque, the most diluted plaque, okay? You isolated a single plaque that you can then continue on to purify for a couple weeks. So when you're doing this, there is a specific type of plaque that we want you guys to pick for the purpose of streaking. The ideal plaque here is being one that is well isolated. The reason we want one that is well isolated is because, again, we're trying to get a single species that we're going to purify over 3 weeks. The more isolated a plaque is from others, the more likely we're only going to pick up one phage particle, one type of phage particle, one species of phage, you could call it. So the more isolated, the better, just to ensure that as we're moving forward with weeks of streaking, we're not accidentally streaking more than one species. So in your lab manual, it's recommended that you pick one that is at least 0.5 centimeters away from any adjacent plaques, and you have rulers in your little lab boxes, so you can actually measure this. But you can also just use your best judgment. One that is as isolated as possible is the best one. And we are going to be using sterile wooden sticks to streak our phage. So let me show you how that works. So sterile wooden sticks are obviously sterile. And what we've learned from this week, hopefully, is that sterile materials have to be worked with in an aseptic zone and handled very carefully not to contaminate all of them. Okay. So sterile wooden sticks are going to be kept in these blue little plastic bags that we do autoclave. Okay. You're going to see tape on them, just like all the other sterile items that are autoclaved. That'll tell you if this is an autoclaved item. You have to be careful with it because it's sterile. So you can't be putting your fingers into the bag and contaminating the sterile sticks. Let me show you how to get those sticks out of the bag. So you're going to be, again, working within the aseptic zone. Your flame is going to be on. You're going to grab your bag. You're going to note that it is sterile with that little blue striped. And then you're going to shake the bag. You're going to shake it until a stick or two poke out of the bag, right? So that you can just grab it without fully going into the bag, okay? And then other ones you can toss back in there and you're good. Use your stick. If you put it down on the lab bench, if you put it down on anything, actually, it's now contaminated, so don't use that particular one. Grab a new stick, okay? I recommend only grabbing sticks out of the bag when you are actively ready to do some streaking. This is not a material that you should be getting out in advance as you're preparing your lab bench. This is something that you get out as you need it. And we'll have plenty around the room for you, so don't stress about having to be like prepared for this. Okay, but be very careful in not contaminating this bag or your sample. Okay. So first thing you're going to do after you've, taken pictures of your direct isolation plates, whatnot, is you're going to get 2 plates. You're going to label the agar side. If this is your first streak, so for most people it is, I always recommend dividing your plate into 3 sections. The one on the left is my preferred streak division on a plate, the T, the classic T, or you can divide into 3 equal parts, whichever floats your boat. I also recommend labeling these sections 1, 2, and 3, 1, 2, and 3, if this is your first time doing this, along with the basic things like your initials, the date, what the material is, streak plate 1, just so you know and you can identify that plate in the future. You are going to, on your direct isolation plate, you're going to pick a plaque, just one. You're going to circle it on the agar side of your dish with a Sharpie, not inside. You're never going to open the plate. On the agar side, you're going to circle it with a Sharpie, the plaque that you are going to be picking from. You're going to put your initials by it, and then you're going to take a photo of this. This photo is going to go into your lab notebook so that if someone is recreating your experiment or looking at your methods, they can look and see your movement through this process. They can see what you started with and what you ended up with. And this is also really good for you guys in your discussion sections of your papers that you're going to be writing, right? This is something that is important that you could talk about. So did you have a certain morphology that you started with when you did your direct isolation plate and then you moved through the streaking, through the purification? Did that morphology stay the same or did it change? And if it changed, why? If it didn't change, why, right? These are all things you can be discussing and things you should be noting, okay? So you're gonna touch your plaque gently with the stick, just once. And I really do mean very gently. There's no need to just like stick it into the agar. Phages are microscopic. You're not gonna see physically on the stick if you're grabbing any, but I promise if you're touching your plaque, you're getting it in there, okay? No need to be aggressive. Then you are going to, using that stick, you're going to streak it into your first section, then your second, and then your third, okay? And I'll show you how to do this in a video, but just let's talk through it verbally, okay? So your first streak should be pretty tight together, right? We're taking from our direct isolation onto a brand new plate. We're just streaking them very close together, okay? Your second streak, you're going to dip into that first section. First, your streak is going to be a little tight, and then you're going to widen it out. And then for your third one, you're going to dip into that second section and then drag it into your third, and you're going to have your really wide streaks. The reason we do that is we want to make sure that we're diluting it as we're moving from the first to the third section. So that means adding more space so that the plaques are more isolated in the third section. Also, you're going to make sure that between each streak, between each section 1, 2, and 3, you're going to be grabbing a brand new stick every single time, okay? Because again, we are diluting it, so that means we are taking an area of concentration, taking a new stick and dragging some of those particles down, getting a new stick and dragging it into another section, okay? If you use the same stick for all 3 streaks, you're not diluting anything. You're just dragging the same sample across your whole plate. You're going to get a very highly concentrated plate, most likely. So let's see how this looks. So I touched a plaque with my stick off to the side. I'm not putting the lid on the bench, and I'm very tightly streaking it very gently all along that first section. Okay, pretty tight. I'm getting a brand new stick. And then I'm diving into that first section and dragging it into the second. And after a little bit of time, I'm widening those streaks out and I'm not going back to that first section. Then I'm getting a new stick. I dive into the second section and drag it into the third. And then with a Sharpie, I am noting on the back of my plate where exactly I stopped with my stick, right? What was the very last point in which my stick connected with that auger? And that's important for later, okay? For a second step, it's very important that you know where you ended your streak. But you'll notice in that video that, again, my streaks started out very tight in my first section, and then they widen out in my third. So after that, that's it. That's the whole streaking process. That's done. But we added we added phage to our plate. Now we have to add bacteria, okay? So we added a little bit differently than we did last week. So you're just gonna take a culture tube. You're gonna add 250 microliters of your Gordonia to it. You're gonna add 3 milliliters of top auger, and then you are going to immediately transfer that to your auger plate, okay? And the way you add it, so you remember how last week we added our top auger to a plate and we swirled it so we can get that top auger all over the plate? Well, we are not doing that this week. And that's a very important reason why, okay? You are going to pour that top auger bacteria solution into that third section, where is your most dilute section, where you have that X written down, okay? You are going to allow that mixture to spread across the plate from the point that's most diluted to the most concentrated, okay? So you're gonna very gently zhuzh your plate, tilt it just a little bit. So you're spreading it, but you're not being very aggressive with it. So the reason that we do that is because we wanna make sure that we're keeping that dilution that we just created intact. So if you pour your auger on the most concentrated section, right, your section 1, and you pour it down into your third, you are essentially moving forcefully those phages, that high concentration of phage that's in your first section, into your third, okay? We want to do the reverse of that. We don't want to accidentally transfer a bunch of phage to what we wanted to be a dilute section, okay? Similarly, if we swirled a plate, we would also be swirling all those phage particles around. We don't want to do that. We are trying to keep our area, our plate, as dilute as possible to keep that dilution that we just created, okay? Let's show us how it's done. So I'm taking my top auger, I'm pouring it directly into that, where that X is. Again, I'm never putting my Petri dish lid onto the lab bench. And very carefully, I'm just tilting it slightly, okay? So it gets as everywhere as possible. If there's a section that you just are not getting, no matter how much gentle tilting you're doing, you can't hit it, that's okay. Just leave it alone. It's better to keep our dilution intact than it is to get like a tiny inch of top auger somewhere, okay? So again, we're not swirling. We are very gently tilting, okay? So let's think of the most important things to remember when we're streaking, okay? One, pick a very isolated plaque, as isolated as possible. Make sure you're getting a fresh stick with every single streak. We're going to be pouring our top auger bacteria solution immediately. No dilly-dallying with that. We're going to pour it onto the auger and not the lid. We're not going to be aggressive at the auger. This is important. You notice when I was streaking in those videos, I was very gentle, right? You could see the lines, but it wasn't like too aggressive, right? You need to be very gentle with the auger. If you think you're being gentle, be even gentler, because I promise you're being a little bit too aggressive, because you think what students typically think is they have to make sure that you can see the line being formed in the auger. You don't. Again, these are microscopic particles. You have no reason that you need to be too aggressive to get them in there. A trick, a practice that I recommend before students come into Open Lab is you just take a pencil and a piece of paper and you very gently just practice drawing very, very faint lines. The amount of pressure that you would apply on your on the pencil that you're holding to get those lines as faint as possible is the same amount of pressure that you need to apply to the stick that you're holding onto the agar. Okay, so very, very light pressure. So let's talk through some scenarios that typically happen. Okay, let's say, like, what happens if you come into Open Lab, you didn't get any plaques in all 3 of your samples? What are you streaking? Okay, well, we are going to give you either an enriched sample that we have created in the lab, or we have some backup direct isolation samples that we are working for you, or you may even borrow another student's plaque. Okay, 3 different options for you. What you're going to do, you're going to take that direct isolation plate, an enriched plate, whatever plate you're streaking from. You're going to do the same thing as I mentioned earlier. You're going to circle a plaque, you're going to write your initials by it, you're going to take a photo so you can put that into your lab notebook. You are going to streak the same plaque twice. So one plaque, touch it once, streak, 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 that's it. And then again, touch the same plaque and do a whole nother streak plate. You're going to do it twice. Same plaque, 2 plates. Okay. If you are borrowing a sample, either from one of the direct isolation ones that the TAs have prepared, from one of the enriched ones that Dr. Ball has prepared, or from your neighbor or someone else in the classroom, it's critical that you get that location information and any other information about that sample for your lab notebook. It is critical throughout this process that you have documentation and photos of the plates, of the history of your streak plates, of your sample. Okay, so if you're borrowing someone, you need to get that information. So someone can recreate that if possible, have all the information, all the history on that particular sample as possible. Okay, this is going to be something that you're going to be told again and again throughout the semester. Make sure you have any background info, location info on any sample you're working with, especially if you're switching samples. So what happens if you do get some, okay? If you do get from your sample some nice little plaques. So if you got, let's say, two different samples, they both had beautiful plaques on them, okay? You are going to do the same thing I mentioned earlier. You're going to find isolated plaque, circle it, put your initials, take a photo, put it in your lab notebook, all that good. And then you are going to take one plaque from your first sample, from one of your samples, do a unique, its own streak plate. Then you're going to do the same thing for a different sample. So you will have 2 streak plates from 2 different plaques from 2 different samples. Okay. If you only got 1 out of 3 direct isolation plates to work, you're going to do the same thing I had mentioned the first time. You're going to take a single plaque and you're going to streak it onto 2 plates. So 1 plaque, 2 plates. And this is in a situation if you only got 1 out of 3 direct isolation plates to work. So what if you were quarantining and didn't come into Open Lab? Or you just weren't able to schedule something, or something went down and you couldn't come into Open Lab last week and you could not do a direct isolation protocol. So what do you do? Well, we're going to borrow someone's. So it's, again, going to be either the direct isolation plaque essays that the TAs have prepared, the enriched ones that Dr. Ball has prepared, or, again, a partner's is always a good one because students, you guys have the most unique samples typically. So we love to rely on each other to move forward with our work. So all 3 options for you, and you're going to do what it sounds like a broken record at this point, but you're going to take a single isolated plaque, you're going to circle it, you're going to put your initials, and then you are going to do 2 streak plates from a single sample. Okay, and again, get location information. If you're borrowing a sample, take a photo, ask them if they have any photos they want to share, anything. Okay, you're going to put all that in your lab notebook. This is essential for your final paper. Please, please, please be proactive now and be getting this information. So pivoting away from streaking things, obviously, if you have any questions about streaking or anything like that, I recommend rereading the sections in your lab manual. They explain it pretty well. And then just going through this material in this slide deck a couple times until you're familiar with it. Okay. But I want to pivot into aseptic technique things. So this was everyone's first week in Open Lab. So some of the stuff might have been new, like this is not muscle memory yet. We're not really sure how to do some of these techniques, but we really need to work on our aseptic technique. Okay. Luna the lab corgi, what she saw this week in open lab, it scared her. She was scared. She saw all the possibilities for contamination and she was really worried. Okay, so moving forward, let's work on these sets of things. Okay, so we're not scaring Luna the lab corgi. One, we're not putting lids down onto our lab benches. Okay, um, your lab bench, even though you wiped it off with Lysol, it is considered a contamination like carnival. There's bacteria there, things you brought in, things that are introduced by some of the various objects in the room. It's contamination. You never want to put anything that is like a shared bacteria tube or top auger lid or your petri dish lid onto that lab bench because it is introducing contamination to your sample. Never put anything down, always hold it. And then work within our aseptic zone. This might be just a lack of familiarity with the flame and working with it, but a lot of people working really far out from their aseptic zone. You have to be able to feel the heat of that flame, and that's how you know you're in the aseptic zone. Obviously not where it's like burning hot, but where it's warm. So just close in with your hands around your flame until you feel it, okay? Make sure that we're passing any glass items through the flame really quickly. I saw a lot of people not doing that with their culture tubes. Anything that's plastic, microcentrifuge tubes, falcon tubes, anything like that, petri dishes, they should be in your aseptic zone, not put into the flame, but enough where we can take advantage of that updraft from the heat to push any microbes away from our samples. And then make sure we are changing tips also. Critical as we're working between different types of samples, right? Sample one, sample two, um, different environmental soil samples, things like that. Make sure that we are changing tips. We're not dipping into like our soil sample, any of our sample things, and then introducing that to a communal bottle of top auger, for example, or a communal falcon tube of bacteria. Um, make sure we're changing our tips and we're taking our time. We're not rushing through things. Mistakes happen when we rush. Take your time, move through the protocol carefully, slowly, just to make sure we get everything right. Okay. So open lab this week is only going to be 1 hour. That means a couple things. One, make sure you're signing up for an hour for open lab for this week. They're going to be for 1 hour for these next 3 weeks because we're going to be doing the same type of protocol. But because it is 1 hour, make sure that we're coming in knowing what we're doing, right? We're going to be efficient. We're not going to sit there not knowing what we're doing. We're going to come prepared. Okay. Hopefully, after this week, we understand the expectations of us in open lab. So we're a little bit more focused and understanding what we need to do before we come in to do it. But obviously, if you have any questions about any of the protocols, anything like that, you can reach out to me. You can reach out to your TA. We're happy to walk through you anything with you so you can come as prepared and ready as possible for open lab. Okay. And make sure we are practicing good aseptic technique to keep Luna the lab corgi smiling the whole time and not horrified by the contamination that she was witnessing. Okay. And then I guess we'll see next week if Luna is a happy corgi. Hope everyone enjoys their streaking.